In the previous lecture, in artificially acquired active immunity, I had stopped at killed vaccines. Now continuing the discussion on artificially acquired immunity, I will take up now live attenuated vaccines, which are different from killed vaccines. Here the term itself indicates live attenuated. Live stands for living. Attenuated means the target organism, that is bacteria or the virus, has lost its virulence. It has been made to lose its virulence, that is disease causing ability, by certain methods. But the bacteria or the virus is alive when it enters the target host, but does not cause the disease because it has lost the ability to cause the disease. Therefore, such vaccines are called as live attenuated vaccines. The first example that needs to be given is BCG vaccine, because that was the first live vaccine that was developed in 1908 by two scientists, Calmette and Guerin, in France. Therefore, it is named after them as BCG vaccine. B stands for bacilli. Bacilli is supposed to be the bacillus, that is the tuberculosis bacteria, which is a bacillus bacteria. Calmette and Guerin are the names of the two scientists who prepared this live attenuated vaccine against Mycobacterium tuberculosis, which causes the tuberculosis. The vaccine was prepared by growing this bacteria in the medium that contained bile salts and human bile. As it is, growing of tuberculosis bacteria in the medium is very difficult. It takes a long time. By growing this bacteria in the bile salts and human bile and aging this culture of the bacillus, that is tuberculosis bacteria, for many generations, in the sense every time you change the media is replenished with the new batch of bile salts and the bacteria is subcultured. And the subculturing method for many generations is called as passages. And this passage term is also used for animal cell culture. When the old culture is rejuvenated by transferring the small sample of that into the new culture, it is called as passage. Like this, by growing the tuberculosis bacteria in many passages or many generations of the culture medium containing bile salts, they were able to attenuate the bacteria, that is, make it lose its virulence or disease causing ability. And that bacteria which lost the disease causing ability is isolated from the media and then used as a live attenuated vaccine for treating the patients who are prone to suffer from tuberculosis. Majorly, this vaccine is used in developing countries like India, Philippines, Thailand, Pakistan, and African countries, where tuberculosis is very predominant. And this is the first breakthrough of live attenuated vaccine which developed in 1908. And it was a successful live vaccine. After that, then another vaccine that came, that is somewhere in 1980s, that is Sabin vaccine. The Sabin vaccine is against the polio, which is also a live virus, polio virus. And this was prepared by growing the polio virus in monkey kidney cell lines and Chinese hamster ovary cell lines. Chinese hamster ovary cell lines are also called as CHO cell lines. And here I have to talk about a terminology called as permissive host and non-permissive host. A permissive host is the one where the virus or the bacteria can cause the disease, whereas the non-permissive host is the one where the virus or bacteria cannot cause the disease but can grow in that non-permissive host without any problem, and that host becomes the vector or a carrier for that virus or a bacteria. Now here I am talking about non-permissive cells. Non-permissive cells of Chinese hamster ovary cell lines and monkey kidney cell lines have been used, which act as the host for the growing of this poliovirus, and the virus, after passages of many generations in this culture medium, that is CHO cell line or monkey kidney cell line, will lose its virulent ability. It becomes attenuated. It is observed that the poliovirus exists in three different strains: A, B and C strain. Therefore, it is quite essential that the polio vaccine, which is generally administered as oral polio vaccine as seen in this slide, which is called as OPV, oral polio vaccine. It is administered orally and it needs to be administered minimum at least for 3 times. Generally, the administration starts at the second month after the birth of the child, as recommended by the CDC, that is the Center for Disease Control, which is United States agency. The Center for Disease Control recommends first administration of oral polio vaccine should be done within 2 months after the birth of the child. The reason being, up to 2 months, the child may be protected by the mother's passive immunity, that is, which has been transferred to the child through the placenta as the child was growing in the mother's womb, and probably it is transferred even through the mother's milk in the form of IgA as a passive immunity, which does not last long. Hence, it is recommended that by the second month, the oral polio vaccine need to be administered. And why 3 times? The reason being, every time when you administer the vaccine, it will start developing the immunity against one particular strain, that is strain A or strain B or strain C, like that. After the administration of 3 times the vaccine, all the 3 strains would have induced the development of immunity in the person. And why orally this vaccine is administered? The reason being, the virus generally infects a person through the oral route because the virus colonizes the intestinal crypts and grows there, and from there it invades the bloodstream and then affects the nervous system. Since the natural route of the virus is through the oral passage, the vaccine, the live attenuated vaccine of polio virus, is also administered through the oral route. Now we will see other vaccines. The measles vaccine. The measles vaccine against the rubella virus is developed in chick embryo or duck embryo. At present, it is also developed in human diploid cells. The chick embryo and duck embryo are non-permissive hosts, and measles virus can be easily grown in them into several passages in several generations, and it will lose its virulence, and then it is extracted from the duck or chick embryo or human diploid cell lines, purified and administered as live attenuated vaccine. Recently, for the typhoid bacilli also, TY21A, a live oral vaccine has been developed, attenuated vaccine has been developed, It is against a strong antigen of the typhoid bacilli. Therefore, it is coded as TY21A. Coming to the pandemic, that is the COVID vaccine against the COVID virus. The Covishield, developed by Serum Institute of India in collaboration with Oxford AstraZeneca, is a kind of attenuated vaccine, but it is not a live coronavirus. Instead, it is a strong antigenic domain of the coronavirus, a protein of the coronavirus, protein surface of the coronavirus, which is cloned in the common cold virus, that is adenovirus of chimpanzee, and that protein is expressed as a strong antigen, hence it acts as a vaccine. But you cannot consider this as a live attenuated vaccine because it can be taken in the example of subunit vaccine. I will be talking about subunit vaccine in few minutes, where a protein domain of the virus is being cloned in another virus, and that virus is attenuated, that is chimpanzee virus, and that virus expresses the protein of the COVID virus, hence acts as a carrier and facilitates the development of immunity in the individual. Since live attenuated adenovirus is used, it can be also given as an example of live attenuated vaccine, but it strongly comes under the example of subunit vaccine because actual COVID virus, attenuated COVID virus, is not used here. Then comes the Sputnik 5. An inactive viral vector has been used by the Russian lab, and that inactive viral vector expresses the COVID protein. And this Sputnik virus, since a live inactive virus was used, it can also be considered the live attenuated vaccine. Now coming to the schedules of the vaccine, it is very important that the vaccines need to be administered at definite time intervals as recommended by CDC. As I have mentioned already, oral polio vaccine need to be administered within 2 months after the child's birth. Similarly, BCG vaccine needs to be administered to the child within 2 months after its birth. Then there are other vaccines like diphtheria, pertussis, tetanus, which is a combined vaccine, which is called as DPT. Diphtheria caused by diphtheria bacteria, pertussis is another bacteria which causes whooping cough, and tetanus is caused by another bacteria. All these three species of bacteria are very deadly. Therefore, it is essential that the child needs to be immunized within 2 months after birth. The DPT vaccine is also done within 2 months after the birth. Then the second vaccination is done on the 4th month of the DPT and OPV. OPV is oral polio vaccine. Hepatitis B may be necessary to be used as a vaccine for some children who are prone to be exposed to hepatitis B virus in endemic areas, but it is not compulsory. Within 16 to 18 months, once again, OPV need to be administered, that is the third dose. And by 12 to 15 months, other vaccines need to be done, that is against the chicken pox, the virus is varicella zoster, as well as against measles, mumps and rubella. Against these, it is called as MMR. This vaccination is a must once again within 12 to 15 months. DPT is also done within 12 to 15 months. So DPT goes through 3 schedules of administration or even up to 4. And OPV also can go up to 4 schedules of administration. Whereas MMR, only 2 schedules of administration, that is within 12 to 15 months once and then within 4 to 6 years once MMR needs to be administered. It is essential that these schedules need to be strictly followed to have a sound immunity in the growing child against these deadly pathogens. People do ignore the later vaccinations, thinking that already the earlier vaccinations have been done. What is the necessity of doing the later vaccinations? But they do not know the advantage of giving these later vaccinations, that is at the 4 to 6 years of DPT, OPV and MMR, because they boost the immunity of the child which has already started developing good amount of bone marrow. At 11 to 12 years also it is recommended diphtheria, tetanus and vis-a-vis. Vis-a-vis is for the varicella zoster administration of the vaccine is essential. So that completes the entire immunization protocol as far as the individual is concerned, who will maintain the immunity against these deadly pathogens all throughout the life in the form of immune memory in the T cells and B cells. And whenever if these pathogens by accident enter the person would be able to fight these pathogens out because the person has been vaccinated, and it is an active immunity. And these live attenuated vaccines will have strong immune response. Now coming to the another type of vaccine, that is toxoid vaccines. The toxoid vaccines are produced from the toxins of the bacteria. Some of the bacteria like diphtheria, tetanus, botulinum and cholera produce toxins which are exotoxins. And these exotoxins are proteinaceous in nature and they have debilitating effect in the individual as soon as they are released by the bacteria once the infection takes place. For example, tetanus and botulinum toxins are neurotoxic. They do cause paralysis of the nervous system once they diffuse into the blood of the individual once the infection spreads. Therefore, it is essential that to prevent the detrimental effect of these toxins if the infection takes place, the toxins can be converted into harmless material by converting them into toxoids. First, to convert them into toxoid, one need to produce toxin in large quantities. Therefore, these deadly bacteria like diphtheria, tetanus, botulinum or cholera can be grown in a medium, and that medium will contain human blood broth. In the case where the broth of the bacterial culture is going to contain human blood, all the requirements that are there in the human blood will be there in the media. Since the human blood is added to the media, it acts like the human environment. The bacteria is inoculated into the media and grown in the incubator by keeping it in the suitable temperature of 37 degrees. The bacteria feels as though it is growing in the human body because you have provided the ingredients that are present in the human plasma or fluid by adding human blood to the broth. As it starts growing, it will start secreting the exotoxin into the media in a copious amount. By growing large quantities of this bacteria in large quantity of the medium, human blood broth medium, one can produce large amount of toxin, and this toxin being proteinaceous, it can be easily extracted from the medium, purified and then converted into toxoid. That is, it is oxidized. Now, how to convert this active toxin which has detrimental effect on the human being into a harmless substance? The toxin is treated with the dilute solution of formaldehyde or phenol or detergent. By treating with formaldehyde or phenol or detergent, it gets converted into toxoid. From toxin to toxoid, it gets oxidized. And toxoid form of the bacterial toxin is harmless, which is later purified, and this toxoid is suspended with aluminum oxide as an adjuvant, which coats over the aluminum oxide particles, or it could be mixed with the adjuvant of lipopolysaccharide of any bacteria, which is harmless. The lipopolysaccharide purified from Mycobacterium can be also used as an adjuvant. And this toxoid preparation is given in an injection, intramuscular injection, as a vaccine. And that also protects against the harmful effect of these bacteria by producing immunity against these toxins, because these toxins converted into harmless form as toxoid are a strong antigen because they are proteins. They trigger the immune response and the antibodies will be produced, and these antibodies will help in tackling these toxins. Now coming to the another part of the vaccine, that is subunit vaccine. What is meant by subunit vaccine? Subunit vaccine is nothing but, instead of using the whole bacteria or whole virus, the antigenic domains of the bacteria or virus can be used to produce the vaccine. The known strong antigenic domains can be isolated. The techniques are available for isolation, and one can identify the genes for these strong antigens in a particular pathogen, and those genes also can be cloned to produce the subunit, which is called as recombinant vaccine. Now the subunit mainly, as shown in the diagram, could be of the capsular origin of the bacteria, that is outside capsule, or it could be a strong membranous protein, or it could be a adhesive molecule of the bacteria which is present in the bacteria on the surface of the plasma membrane, which gives adhesive ability, that is to attach to the cell walls. Or it could be pili. Pili is cytoplasmic projection of the bacteria, which is seen in the bacteria, which is used for the conjugation purpose by the bacteria or also for the attachment. Then bacteria, some of the bacteria contain flagella, and the flagella is made up of a protein called as flagellin, which becomes a strong candidate as an antigen for the vaccine production. The exotoxins, we have already spoken about it as toxoid. Exotoxins produced by the bacteria, also the strong candidates for the vaccine. The lipopolysaccharide part of the bacteria also could become the vaccine candidate. So, using different parts of the bacteria or different parts of the antigenic domains of the virus is called as subunit. Now here an example of Salmonella typhi, which causes the typhoid fever. This bacteria has three strong antigens. The H antigen belongs to flagellin protein, that is of the flagella. The O antigen belongs to the membrane protein, which is another strong antigen, and the capsular antigen which is a polysaccharide, which is called as VI antigen. And these three candidate antigenic determinants or epitopes have been taken into advantage to produce vaccines against these antigenic domains of the Salmonella typhi. Coming to the coronavirus, which caused the pandemic, which has large number of antigenic determinants, and these antigenic determinants have been exploited for the production of vaccine as subunit vaccine. Almost like about 157 companies and 157 different types of vaccines have been attempted against the coronavirus. Some succeeded and some failed. The strong candidates for the production of vaccine, subunit vaccine for the coronavirus are the spike protein, the membrane protein, the envelope protein, nuclear capsid protein, the genomic RNA as RNA vaccine, and the lipid bilayer part of the envelope protein. All these candidates have been exploited for the production of subunit vaccine against the coronavirus. In University of Science, Bangalore, they produced a heat-stable vaccine against the coronavirus, that is COVID-19, which is a subunit vaccine containing the spike protein. The spike protein is the one which is shown here with the red color, spike protein. And this antigenic domain, which is a strongly antigenic, by identifying the genetic component responsible for the production of this spike protein, they were able to use that gene and clone it in animal cell vector, that is viral vector or yeast cell vector, and grow that vector successfully in animal cell or yeast cell to produce the subunit spike protein in large quantities. The protein produced in the cell culture is isolated, purified, and used as a subunit vaccine against the coronavirus. And it is heat stable and can be stored in the laboratory temperature. That is the advantage of this vaccine. Coming to the polysaccharide vaccines, these are the polysaccharides that are present in the capsular region of the bacteria, and they could be of two different types of polysaccharides. One group of polysaccharides could contain glycosaminoglycans, that is amino sugars, which are found to be the strong antigenic determinants, glycosaminoglycans. The other type of polysaccharides are the pure polysaccharides, which do not contain glycosaminoglycans. Those polysaccharides which contain glycosaminoglycans, that is amino sugars, are strong antigens, whereas the other polysaccharides which do not contain glycosaminoglycans are not strong antigens. In that case, such polysaccharides may need assistance from another molecule, which we will discuss little later. Here, the polysaccharide vaccines against the Haemophilus influenza type B or Streptococcus pneumoniae or Neisseria meningitidis, and all these capsular polysaccharides were found to contain glycosaminoglycans. Therefore, they are strong antigens and vaccines could be easily produced using these subunits. This also comes under the subunit vaccine because it is not a whole bacteria. It is the component of the bacteria that has been used for the production of vaccine. Now coming to the other polysaccharide vaccine, which I mentioned that these polysaccharides do not contain the amino sugars. Therefore, they are conjugated with the known strong antigens, which acts as facilitators, carriers, and help in production of vaccine against the weak antigen also. But there is a dual advantage here. Say for example, O antigen of Salmonella typhi is a poor antigen. It doesn't have good antigenic ability because it is a pure polysaccharide. Therefore, it is conjugated with tetanus toxoid, and tetanus toxoid is a protein and it is a strong antigen. When it is conjugated with tetanus toxoid and injected into an individual, the individual will produce two types of antibodies, one against tetanus toxoid, which is a strong antigen, and other against the O antigen of the Salmonella typhi, which is a dual advantage. Similarly, VI antigen of Salmonella typhi is conjugated with B subunit protein of E. coli toxin or with exoprotein A of Pseudomonas aeruginosa. Both these two proteins, that is B subunit of E. coli toxin or exoprotein A of Pseudomonas aeruginosa, are strong antigens, and VI antigen is a pure polysaccharide. On conjugating with this, it will help in production of antibodies against both the components. Same way against Shigella O antigen, which is a gastrointestinal pathogen, exoprotein A of Pseudomonas aeruginosa is conjugated with Shigella O antigen. Same way, E. coli O antigen is conjugated with tetanus toxoid. So advantage of conjugating with a strong antigen, which is also can be considered as a hapten carrier. And that strong antigen, as it is capable of producing the antibodies against it, because it triggers the immune response very well. And in the process, it also triggers the immune response against the weak antigens, which otherwise would not have been able to induce the immune response on its own. Now, some special proteins of the bacteria, which I have already mentioned, like flagellin of Salmonella typhi, pilin, which is derived from the pili of the Neisseria gonorrhea, porin 1, porin 2 of Neisseria gonorrhea. Porin 1 and porin 2 are the adhesive proteins, are also a good candidates for the production of vaccines, and they also come under the subunit vaccines. Another subunit vaccine that is produced is by using attenuated Salmonella typhi for the production of genetically engineered cholera toxin. So the genetically engineered cholera toxin carrying plasmid is introduced into the Salmonella typhi, which is attenuated, and it is orally administered. Salmonella typhi colonizes the intestinal tract naturally. Since it is attenuated, it cannot cause the disease. It will remain in the epithelial lining and grow there, and as it is growing there, it will now produce genetically engineered cholera toxin, which has lost its virulence. The genetically engineered cholera toxin is the one which is produced by modifying certain genetic codes in the gene of the toxin, and these genetic codes were responsible for some essential amino acids which exhibit the toxicity in the toxin. By modifying these amino acids, by modifying the genetic code, the toxin was made into harmless substance, and it is attenuated toxin, and was made to produce in Salmonella typhi. Now here is a dual advantage. On giving attenuated Salmonella typhi carrying genetically engineered cholera toxin to a person, the person will produce the immunity against Salmonella as well as against the cholera toxin. It's a dual advantage. Now coming to subunit vaccines produced by recombinant technology. The recombinant technology is nothing but viral vectors or plasmid vectors are used to carry the gene of choice of a strong antigenic domain or antigenic determiner of a particular pathogen. By cloning this gene in the suitable vector and a suitable host, one can produce large quantities of that subunit antigen in the medium, and which can be purified and administered as a vaccine. So here the genetic engineering technology has been used. Now what are the vaccines that are produced by recombinant technology? That is against hepatitis B antigen, influenza virus, herpes simplex virus antigen, that is a glycosaminoglycan protein, rabies vaccine, measles vaccine, Salmonella typhi antigen, meningococcus, and Haemophilus influenza. As far as rabies vaccine is concerned, earlier rabies vaccine was produced by giving the rabies virus as an injection to a non-permissive host, that is sheep, by injecting that virus in the brain of the sheep. And the virus is allowed to grow in the brain of the sheep in the cerebrospinal fluid for quite some time. Being a non-permissive host, the sheep does not succumb to rabies. The virus, after reaches a good titer, they used to extract the virus from the cerebrospinal fluid of the sheep and then purify this virus and heat kill it or chemically kill it and administer as a killed vaccine. Unfortunately, sometimes due to improper purification process, it was observed that in some instances the rabies virus which was extracted from the sheep brain and purified would contain some part of the sheep brain protein with it, and this would cause some complications in the people who were administered the killed vaccine of the rabies, and it led to a disease called as encephalomyelitis. As the antibodies were developed against the rabies virus, which is killed rabies virus, there were also antibodies against the brain proteins of the sheep, which was not properly isolated, resulting in the cross reactivity with the human individual brain protein, causing a disease called as encephalomyelitis. Therefore, it is absolutely essential when the vaccines are produced in animals and purified. Stringent measures have to be taken in the purification process where the animal components, proteins, should not accompany the vaccine, which leads to complication. So, coming to the vectors that have been commonly used for the production of the vaccines against the listed organisms or listed viruses and bacteria. as shown in the slide. The vectors were vaccinia virus, adenovirus, canary pox virus, and poliovirus. In the non-virulent form, these viral vectors were used for the production of vaccines against different pathogenic organisms, especially the pathogenic viruses. And these vaccinia virus, adenovirus, canary pox virus, and poliovirus are used for infecting animal cell cultures. And they carry the gene of choice of the antigenic domain of a particular virus, and that gene of choice will express in the host cell, that is animal cell culture, and after sufficient expression and sufficient titer of that protein being expressed or the antigenic domain being expressed, the protein is harvested from the cell culture, purified and administered as recombinant subunit vaccine. The commonly used bacterial plasmids are PUC1 and PBR322, as well as yeast also is used with its own plasmid as a vector for the production of viral subunit vaccines as well as bacterial subunit vaccines. Here is the diagrammatic representation of the plasmid cloning vector of the bacteria, and you know that the site A and B that is shown is the multiple cloning site, which is called as MCS, which has on either side the restriction sites, and it is a large segment which can accommodate a large gene segment of the choice of the viral antigenic gene or bacterial antigenic gene, which can be cloned in this part where A and B is shown in the diagram, and this plasmid vector can be grown in a suitable bacteria and produce the subunit vaccine in large quantities. Here is an example of production of subunit vaccine against the hepatitis B virus subunit antigen. The antigen of choice, that is strong domain of the hepatitis B virus, is chosen, that is shown on the extreme left hand corner as a gene segment, and which has got the restriction site on either end as EcoR1, and the yeast vector is used for cloning this gene, and the yeast vector has the alcohol dehydrogenase as the marker gene with its own promoter, ADH promoter, and it also has a marker gene for the production of tryptophan. The hepatitis B antigenic gene is cloned in the yeast vector in the segment that is shown in the diagram and made to express in the yeast cells, which are grown in tryptophan deficient media. The tryptophan required for the yeast to grow is provided by the gene that is present in the plasmid, and the cells which have not been transfected by the plasmid will not survive in the media, because the cell, if it does not carry the gene for tryptophan, the mutant yeast cell which is lacking tryptophan will not survive. The cells that survive in the media, the mutant yeast cells, which are tryptophan deficient yeast cells, which survive in the media, that is tryptophan deficient media, are provided with tryptophan because of the tryptophan gene being present in the plasmid, which produces the tryptophan under the command of the promoter of alcohol dehydrogenase promoter. As the alcohol dehydrogenase promoter promotes the expression of hepatitis B antigen as well as tryptophan, those cells grow in the media and those cells are harvested because they are known to contain the plasmid which is carrying the HBsAg, that is hepatitis B antigen. And those cells will have produced the protein, that is hepatitis B antigen protein, in large quantities in their cytoplasm. On harvesting these cells, the cells are lysed, and then the protein of choice, that is hepatitis antigen, subunit vaccine antigen, is isolated, purified, and this is another example of recombinant subunit vaccine production. Here is a diagrammatic representation of the 157 vaccine candidates as far as coronavirus is concerned. It could be the RNA, or it could be the DNA produced from the RNA of the virus by cDNA technology, and live attenuated vaccines of the coronavirus, or inactivated vaccines, or cloning the particular antigenic domains in the viral vectors, like that. About 157 vaccine candidates were identified and were tried for the production. But how many succeeded, we are not sure, but those which succeeded are as follows. The subunit vaccines against coronavirus, that is COVID-19, one of them is Novavax, a plasmid vector recombinant protein, a subunit vaccine produced by Serum Institute of India. A recombinant protein is nothing but it is a protein unit of the virus. It is probably the spike protein of the virus, which is used as the candidate, and the gene for that was cloned in the plasmid vector, and Novavax is the trade name of the vaccine. Jensen is another trade name for the viral vector protein subunit produced by Johnson & Johnson of USA. ZCoV-D is a DNA plasmid protein subunit vaccine produced by Zydus Cadila. And Covovax was produced by Biological E, which is another industry that produced the recombinant subunit vaccine against the coronavirus. Now coming to another type of vaccines, that is DNA and RNA vaccines. So DNA vaccines are the gene of choice against a particular antigenic determiner, which is cloned in a suitable vector. And that particular gene in the form of DNA would express in the target host after injecting the DNA as a vaccine directly. And that DNA which has been identified, which is the actual gene of a particular antigen, is coated on the gold nanoparticles and inserted or transferred into the muscle cells of a target host by gene gun approach, which is called as biolistic approach. And gold nanoparticles act as carriers for that DNA. And then that DNA expresses in the target host, and that antigenic protein produced by that particular DNA through mRNA route will serve as subunit vaccine. There is also mucosal DNA vaccine against Vibrio cholera and Shigella species, and a vaccine was also being developed against the coronavirus. Here, the DNA of a particular antigen is administered through the nasal route by taking advantage of the nasal mucosal passage. It is directly sprayed into the mucosal passage of the nasal route, and the mucosal passage absorbs this DNA, which is coated over some nanoparticle, and then the expression of that antigen in the form of protein takes place, giving rise to protection. mRNA of the pathogen for a particular domain can be also used as a vaccine, and this mRNA of a particular antigenic domain responsible for a particular dominant protein which is acting as an antigen can be transferred by coating this mRNA on to gold nanoparticles by using gene gun approach or bialistic approach, similar to the DNA which has been transferred into the muscle cells. Here also mRNA can be transferred directly into muscle cells. One precaution need to be taken that unlike DNA, mRNA is unstable. DNA is quite stable, mRNA is highly unstable. Therefore, it may undergo breakdown when it is introduced into the muscle cell of the target store. Therefore, it needs to be protected against the digestion by the RNAs by using special proteins which prevent it from digestion by binding those proteins with the mRNA. But these proteins will not prevent this mRNA from being expressed in the host cell protein synthesis apparatus for the production of the antigenic domain that is required as an antigen as a subunit vaccine. So RNA vaccines against the coronavirus have been successful, such as Moderna COVID-19 produced by Moderna lab. Similarly, a liposome nanoparticle based mRNA vaccine produced by Pfizer BioNTech in USA. These two vaccines were administered and they were successful in United States as far as RNA vaccines against the coronavirus is concerned. Then comes the multivalent subunit vaccines. Here, when you talk about multivalent, it is with response to antigenic domains. Different antigenic domains or different antigenic determinants can be combined together to produce different antibodies against these antigenic domains by coating these antigenic determinants on a solid matrix, which is an inert solid matrix. And this inert solid matrix could be a liposome. A liposome could be a carrier which may carry these antigenic determinants within itself, or it could be made to carry these antigenic determinants on its surface. And they are called as ISCOMs. ISCOM stands for immunostimulating complexes. And they are predominantly liposomes which help in carrying these antigenic proteins of different antigenic determinants within themselves or on their surface and facilitate as vaccine carriers of subunit vaccine and help in developing antibodies against these antigenic determinants in the host cell. And these could be administered as injections, intramuscular injections, and some of the liposomes which are very small enough can be also used or can be administered as intravenous injections. Depending on the antigenic characteristics and the liposome characteristic, the administration is varied, and they also help in production of antibodies against the multiple antigenic determinants at the same time, helping in developing the immune response of a person. This is how artificially acquired active immunity can be achieved in an individual by using different methodologies for the administration of killed vaccines, live vaccines, as well as subunit vaccines. This is all about the acquired immunity in a person.